There's nothing more of a nuisance than a mosquito bite. Well, besides when they buzz you in the ear while trying to sleep. But believe me, if you happen to get dengue fever from one of those bites, not even making the cross on the wheel will help you. Dengue fever is caused by the dengue virus transmitted by mosquitoes. The disease is characterized by high fever, bone and joint pain, as well as pain in the back of the eyes. In most cases, it turns out asymptotic, or with mild or sometimes incapacitating symptoms without any further complications. But, on occasions, the disease can become way more serious, causing internal bleeding and even shock. Dengue fever is thought to originate in Asia, or probably Africa about 2000 years ago, where it jumped from monkeys to humans through mosquitoes, finally establishing as a consolidated urban cycle involving peridomestic mosquitoes. The ancestral sylvatic cycle involving monkeys and canopy-dwelling mosquitoes still persists up to now in West Africa and Southeast Asia, and numerous cases of recent outbreaks have been associated with spillovers from monkeys. Brought during the slave trade, dengue fever in the Americas is quite recent but widely spread, having a greater prevalence than in Africa or even Asia. And, although dengue virus has been found either directly or indirectly in monkeys in South America, there's no evidence of a sustained outbreak causing sylvatic cycle involving non-human primates. However, it is important to determine whether a dengue virus sylvatic cycle is emerging in the Americas, which will have important public health implications. Besides, this could explain some rather mysterious outbreaks in remote areas of South America. There is scattered evidence suggesting that a sylvatic cycle for dengue virus involving several species of mammals may exist in the Americas, and one of the main non-primate candidates are bats. But why bats? Wouldn't it be better to search in animals that are phylogenetically closer to us? Well, yes, in principle, it is more likely for a virus to jump from one species to another if these are closely related. Dengue virus, for example, has been found to surely infect at least 6 species of mosquitoes and 7 species of primates, including us, but also has been found in birds, bovines, dogs, horses, pigs, and other small animals, so viruses are not so picky about hosts. In addition, viruses have very high mutation rates, which allow them to quickly form quasi-species with the potential to infect new hosts. But the main reason really is that bats are known to harbor thousands of different viruses, many of which have jumped to humans. Some of these include Hendra virus, Ebola virus, the COVID-19 causing SARS-CoV-2, and who knows how many more are yet to come. Approximately 75% of the diseases that have emerged during the past 2 decades have wild reservoirs, and in this sense, bats are host of high viral diversity with high zoonotic potential worldwide. In fact, dengue virus and other flavivirus have been found in bats previously. However, evidence suggesting that bats carry and presumably sustain dengue virus infection is still inconclusive. Today, I'm going to talk about 2 papers that we published quite a few years ago, where we asked Nature if there are bats infected with dengue virus in areas of high prevalence in Mexico, and if this is the case, if these bats are amplification hosts or terminal hosts. So, our first approach was to capture bats from 2 states of Mexico with contrasting ecological characteristics but concurrent dengue virus transmission in humans, and determine if they are or have been infected with dengue virus. This research was conducted in 2012, and at that time, Mexico had about 50,000 cases and ranked as the 12th country with most cases worldwide. The most affected states were Veracruz, followed by Morelos, Yucatan, and Guerrero. However, this is the number of cases. If we look instead at the incidence, which is the number of cases per 100,000 inhabitants in 2012, the state of Morelos jumps out of the scale. The number of cases per municipality in both sample states, Morelos and Campeche, was a pain to get. And actually, this is a reconstruction of the number of cases I came about after a really tedious search in archived news and partially accessible official reports. Therefore, it is anything but accurate. Nevertheless, the ecological characteristics of the sampling sites in Morelos, here marked as blue dots, consisted of urban and peri-urban areas in Jojutla and neighboring municipalities. Cuernavaca and Cuautla at the top had way more dengue cases, but these are big cities where no many bats are found. Contrasting to the state of Morelos, the sampling sites for the state of Campeche consisted of rural areas with fragmented tropical deciduous forests. In addition, and unlike the state of Morelos, there are monkeys in Campeche, which might increase the chance of the establishment of a sylvatic cycle. To capture the bats, we used mist nets. These nets have very fine threads that are unhearable to the bats' echolocation navigation mechanisms, so the bats just fly straight to them without even noticing. The nets were opened at sunset, and the bats captured in the span of 4 hours were placed on cloth bags and taken to the camp facilities located in both states. We collected both males and females, but released the pregnant and nursing ones. In total, considering both sampling states, we caught 240 bats belonging to 3 families and 18 species. The most abundant was the fruit-eating bat Artibeus jamaicensis. They are everywhere, and only one bat escaped our identification skills. Notably, we caught 2 vampires from the species Desmodus rotundus. Those guys are super chunky, and are the only bats that can gallop to chase their prey. Like a lion hunting a buffalo. It's so funny. Anyways, the same night after capture, back at the camp facilities, the bats were anesthetized with pentobarbital, bled by cardiac puncture, and subjected to necropsy to assess any abnormality like the presence of petechia in internal organs, which would indicate internal bleeding. During necropsy, the liver and spleen were extracted to determine the presence of dengue virus RNA as indicative of an active infection, and to determine the presence of anti-dengue virus neutralizing antibodies as evidence of past infections, the blood was centrifuged and the cell component discarded. All samples were frozen in liquid nitrogen immediately after extraction for transportation to the lab, where the molecular analyses were conducted. The necropsies showed no indication of hemorrhages of apparent pathological signs in any bat, except for an Artibeus jamaicensis adult female that had what appeared to be a tumor in one mammary gland. Once back to the lab, RNA was extracted from the tissue samples by the Trisol protocol, and the presence of dengue virus RNA was assayed by PCR. Water, instead of the extracted RNA, was used as a negative control, and RNA extracted from suckling mice brains inoculated with dengue virus were used as positive controls. Furthermore, to evaluate the quality of the RNA extractions, the housekeeping GADPH gene from bats was also amplified in parallel. To control for the presence of inhibitors of the polymerase chain reaction, the tissue samples were spiked with dengue virus propagated also in suckling mice brains before the RNA extraction. This was really important since working with strange wild animals is a totally different story than working with standardized biological models. And who knows if bats have some sort of strange protein or something that causes the PCR to fail. A quick side note. Yes, you heard well. Virus propagated in suckling or neonate mice brains. This is super heavy stuff, and forgive me for the language, but this shit has to end. The thing is that we don't have an efficient way of producing dengue virus in the lab. Growing virus in cultured cells takes time and effort, and not much virus is recovered. Adult mice do not get infected well or at all, except for those so-called humanized mice, which are severely immunocompromised and grafted with human cells. So one of the only options to produce a lot of dengue virus is to use baby mice to which the virus is inoculated directly into the brain, and then extracted after the virus multiplies there. However, we have abandoned this method of producing virus for several years now, and we do not have intentions to use it again unless strictly necessary. We prefer to spend a little bit more resources, time, and effort in growing virus in cultured cells. Anyways, the PCR analysis failed to show evidence of active dengue virus infections in the tested organs, while the dengue virus spiked tissues showed the expected amplicon even after 100-fold dilutions of the extracted material. So what we had left to do is to test for previous dengue virus infections in these bats. For this, we used the plasma of the bats to test for neutralizing anti-dengue virus antibodies by the plaque reduction neutralization test. This is really the gold standard for testing the presence of antibodies produced by an organism, and allowed us to determine if the bats have previous encounters with the dengue virus despite not having an active infection at the moment of capture. The assay consists of mixing the bat plasma with dengue virus and incubated for 60 minutes at 37 degrees Celsius. Then the plasma/dengue virus mixture is added onto a confluent monolayer of African green monkey kidney cells cultured in 96 well-cultured plates. The inoculum was then removed after an hour, the cells washed with PPS, and culture medium added to incubate further for 24 hours. The idea of this is that if the bats had a previous encounter with dengue virus, then the produced antibodies will bind to the virus and neutralize them, rendering the virus incapable of infecting the cultured cells. So, after incubating, the cells were fixed with ice-cold methanol, and the viruses that were able to infect the cells were detected with a double antibody system that recognizes the E protein of the virus and amplifies the signal through a colorimetric reaction. Basically, the envelope protein of the virus within the cells is detected by a mouse anti-envelope protein antibody, which in turn is detected by a second anti-mouse antibody that has an enzyme coupled to it that converts a molecule into a dye. So, if the wells do not become colored, it means that the bat serum contained anti-dengue virus antibodies capable of neutralizing the virus. As controls of the PRNT, we used one serum that tested negative and another one that tested positive for anti-dengue virus antibodies. In addition, we also used the anti-E monoclonal antibody produced in mice. However, none of the plasma samples showed neutralizing activity against the dengue virus, as well as for the negative human serum, whereas the positive human serum and the mouse monoclonal antibody showed a very noticeable neutralization activity. As a side project, and since we were drawing blood samples from the bats, we also had the question if the bats that resulted positive for dengue virus, if any, had difference in their hematological values. So we spent entire nights on the camping facilities, counted red blood cells, all types of leukocytes, platelets, and all sorts of measurements. Unfortunately, since none of the bats resulted positive, there was nothing to report. One of the bats apparently was infected with some sort of microfilaria though. So we didn't find dengue virus RNA nor anti-dengue virus antibodies in the captured bats. However, this does not suffice to conclude that bats are not reservoirs for dengue virus. It could be possible that the bats are less likely to be captured if infected by dengue virus. Perhaps they feel sick and don't leave their roosting sites until healed or death. Another possibility is that bats become seronegative after a while, or clear the virus infection in the absence of neutralizing antibodies. Furthermore, 240 bats aren't that much really. It could simply be that we didn't were lucky enough to capture an infected or previously infected bat. After all, the chances might be really small. For example, sylvatic dengue virus has been maintained in non-human primates in Malaysia without detection for nearly 4 decades. So these studies should be extended probably indefinitely, as a long-term surveillance program, really. It will be nice to just take blood samples and free the bats afterwards though. This way, no more bats have to die for the cause. Nevertheless, capturing and testing wild bats for dengue virus wasn't enough for us, and such long-term surveillance programs have not been established for bats yet. So, to push the investigation further, we decided to capture some bats, take them to the lab alive, and inoculate them via different routes with dengue virus to see if they become infected. We captured the bats in the municipality of Huejutla in Morelos, where we previously captured the bats for the previous paper. We went for Artibeus jamaicensis bats because this species is widely spread. It was the most abundant in the locations we chose to sample, and because it roosts and feeds in proximity to humans. Moreover, evidence for dengue virus infection in wild Artibeus bats has been reported previously in other studies. Okay, so the bats were enclosed in cages built with half an inch PVC pipes, joined by 3-way PVC elbows to form a 1.7 cubic meter cage. This cube was then covered with shade netting, and a couple of small apertures were cut on the top side to fit the bats. We then glued some Velcro on those apertures to easily open and close them daily and introduce all sorts of fruit threaded on skewers which we hanged from the top of the cage. After capture, the bats were brought to the lab that same night and were left in quarantine for 1 week before beginning the experiment. Before their enrollment, the bats were tested a couple days after arrival and the day before the experimental inoculation for dengue virus infections by PCR. And once a carryover infection coming from the wild was discarded, the experiment began. We performed the experimental inoculations in 4 different occasions. For the first one, we captured 15 bats. 10 were destined to be inoculated with dengue virus, while the other 5 were mock inoculated. The first round of inoculations were injected subcutaneously in the chest as an attempt to generate an initial localized infection, as the dendritic cells and macrophages resident to the skin are supposed to be the first cells to become infected after a mosquito bite. Parallel to the bat inoculations, we also inoculated suckling mice in the brain to test the viability of the viral stock, which in this occasion was propagating in a mosquito-cultured cell line. The way we proceeded to test the infections was to take blood samples by venipuncture of the cephalic vein from a pair of bats and to euthanize them the next day to extract the whole blood by cardiac puncture, do necropsies, and then extract the spleen. This way, each day we will obtain either a blood sample or the whole carcass until no more bats were left. The spleen and blood samples were used to test for dengue virus RNA by PCR, as explained previously, but the blood was also tested for dengue virus antigens, specifically the NS1 protein produced by the virus and which is secreted to the plasma, and for anti-dengue virus antibodies produced by the bats. This was done using enzyme-linked immunosorbent assays. Briefly, for the detection of NS1, we used the Platelia dengue's NS1 kit, which is a one-step sandwich immunoassay that uses antibodies against NS1 that bind this antigen to a microplate well on one side, and another anti-NS1 antibody coupled to horseradish peroxidase on the other side. This horseradish peroxidase catalyzes the conversion of a non-colored substrate to a colored product. So, if the microplate well becomes colored, it means that there was NS1 in the plasma sample of the bats. For the detection of anti-dengue virus antibodies produced by the bat, we used the PanBio Diagnostics Dengue IgG Capture ELISA. This assay works by detecting human antibodies that bind to a dengue virus antigen provided in the kit. Since this kit didn't bind bat antibodies, instead we set up a competition ELISA where the bat serum was mixed with the dengue antigen provided in the kit before the addition of the horseradish peroxidase coupled anti-dengue virus tracer antibody. While this was reacting, we added into the assay plate the human serum positive for dengue virus provided in the kit, and then we added the bat serum /antigen /tracer antibody to the assay plate and read the amount of color produced after several washes to eliminate unbound antibodies. So, if the bat had anti-dengue virus antibodies, and since this can't be recognized by the assay, no color should be produced because the antibodies provided as positive controls wouldn't have any antigen to bind to because it already reacted with the bat's antibodies. Anyways, with this first batch of bats, we didn't saw any positive result except for one bat, bat number 8, that was sacrificed on day 7 post-inoculum. However, the PCR band produced had a smaller molecular weight than the expected, and the ELISA assay showed levels just barely above the detection threshold. Given these results, we decided to get another batch of 12 bats and inoculate 8 of them, but now intraperitoneally to increase the virus absorption by the tissues. The rest of the bats were again mock inoculated with cell supernatant without dengue virus. The results were the same. We got some spurious bands in 3 of the bats, and a very faint signal in the NS1 test. By the way, all the suckling mice inoculated in parallel showed signs of viral infection and the tests resulted always positive. Since we don't know the possible kinetics of a dengue virus infection in bats, we did a 3rd batch of inoculations, but extending the follow-up time from 9 to 12 days. In addition, the blood extracted from the bats was immediately used to feed mosquitoes with artificial feeders. These feeders have an inverted cup to hold the blood and allow the mosquitoes to feed through this parafilm. Surrounding the cup, there is a jacket in which warm water is recirculated using a water heater circulator to keep the blood warm. Again, none of the bats resulted positive for the test with only one spurious band. The same was for the mosquitoes, none of them became infected. One bat-mosquito couple showed a high colorimetric reaction in the NS1 test. However, this bat was sacrificed on day 1 post-inoculum, so it is possible that we detected the NS1 coming from the initial inoculum, which was also eaten by the mosquito. On day 4, This same batch of bats showed a strong signal in the NS1 test, but failed to show any traces of viral RNA. Secretion of NS1 into the plasma usually comes along with increases in the viremia levels in patients. Thus, in the absence of any evidence of viremia, these results are difficult to interpret. Okay, to finalize, we only had one more thing to test, and that was a more natural way of inoculating the bats using infected mosquitoes. It is known that the mosquito saliva enhances dengue virus infection. Therefore, we first infected mosquitoes with dengue virus using these artificial feeders, and then make these mosquitoes bite the bats. On day 9 after mosquito infections, and after confirming they were indeed infected, the bats were exposed to the hungry mosquitoes by placing them in mesh bags and then into the mosquito cages. This time, we extended again the experiment to 17 days, giving more time for the virus to get going in bats. But once again, none of the bats became infected. We didn't detect any viral RNA, neither had we detected NS1. Furthermore, the bats didn't produce anti-dengue antibodies. We sequenced all the suspicious PCR bands obtained in all of the 4 experiments, but only their spurious nature was confirmed. The necropsies showed nothing, and no clinical signs were observed in the inoculated bats. They didn't change their behavior or feeding pattern. We tried to measure the bat's temperature with an infrared thermometer, but this turned out difficult to interpret because bats are rather strange in this regard. They had been described as having the purest type of mammalian thermal regulation, according to Stone and Wiebes. However, I will regard them as expert thermoregulators, because their temperature can range between 3 degrees Celsius to 41 degrees Celsius, according to their needs and environment. These guys practically present temperatures almost equal to ambient temperature while at rest, but when active, their temperature rises almost to 41 degrees Celsius. They have to do this because they have a really high surface area to mass ratio, meaning that they lose heat very fast, but at the same time, they are the only mammals capable of flight, which demands really high metabolic rates, so they really can't afford to be warm if not strictly needed. So we also did hematological measurements like in the previous paper, but again, we didn't see any clear pattern in their hematological values. So, in conclusion, wild bats don't seem to be reservoirs of dengue virus, and captive bats are not susceptible to the virus. However, this study sampled just a handful of bat species, and only one was tested experimentally. Given that bats are the second most diverse family of mammals, there's a lot of species to sample and test. Our results are in agreement with others that have performed experimental inoculations in different species of bats, but this contrasts with many other studies reporting dengue virus infections in wild bats. All these results are not easy to reconcile, but the tendency is to classify bats as not reservoirs of dengue virus. The current evidence of dengue virus infections in wild bats is based on serology or PCR tests. But the thing here is that there is a lot of cross-reactivity among the Flaviviridae family of viruses. On the other hand, we still lack a lot of the knowledge about bat biology and ecology. So the possibility that conditions in captivity fail to fully reproduce the conditions in which bats encounter virus in nature cannot be excluded. And if it is found that bats can't actually harbor dengue virus, then it might be interesting to ask why. If bats are reservoirs of hundreds of viruses, then why they are refractory to dengue virus? If we found something about this, it might turn useful to develop treatments against dengue virus. Another interesting question is, if it turns out that bats are actually reservoirs of dengue virus, then what are the implications? In one hand, bats can eat thousands of mosquitoes each night, greatly diminishing the mosquito populations. It is for this reason that some towns actually promote bats to be near houses by making them artificial roosting sites. But on the other hand, if bats have dengue virus, and they are near or even in the houses, then what's the overall balance? What will win? Bats spreading dengue virus to humans through mosquitoes, or bats preventing the spread of dengue virus by eating the mosquitoes? As a side note, I just came into a paper that proposes that bats can become infected with dengue virus by eating the infected mosquitoes. So, this is becoming interesting. All in all, I had a lot of fun participating in this research. and I learned a lot. Oh, and I had to receive 5 shots of the rabies vaccine because I got bitten by the bats on a couple of occasions. That wasn't fun. So please do not manipulate wild animals if you are not trained to do so, and don't mistreat or hate bats. They are not to blame for anything and are quite important for the ecosystem. If, after hearing my robotic and poorly articulated voice, you are still here, I just want to mention that this video is actually the odd one. Most of the future videos in this channel will be more like hands-on on the table doing experiments. Like this one I'm preparing about the grease reaction. The thing is that when we were working on this research, I didn't know I would end up making these videos, so I didn't take any footage. Anyways, thank you for watching.
If you liked this video and want to support the channel, please share.
I allowed them to bite me several times, but I'm not getting any wheel.